La doctora Dalila Pinto ha tenido un viaje un poquito accidentado, pobre, pero por fin ha podido llegar directamente desde Nueva York y va a hablarnos, ella es la directora del laboratorio genético de Mount Sinai, que trata sobre el autismo, que estudia la genética en el autismo, y va a hablarnos de estos aspectos y las últimas técnicas y descubrimientos. Gracias, Dalila.
All right. Good morning. Still good morning, right? It is for me. Okay, so this is going to be in English, so I hope that's fine.
Yeah, perfect.
My Spanish is not perfect. All right. Although I'm Portuguese, and my Portuguese is not perfect either anymore. All right. Oh, okay. Perfect. Okay. So thank you very much for the introduction. So I planned my presentation in two parts. First, I will give you an overview of the studies that we are conducting on autism, so autism spectrum disorders in general. And then I will also show you an overview of the work that we have been doing on Landau-Kleffner syndrome, which is a much more rare syndrome compared to autism spectrum disorders, but some individuals with autism can also have LKS, and some individuals with LKS can have autistic features. All right. So, autism spectrum disorder, as the name says, is really a heterogeneous spectrum group of disorders that really impact mostly two domains, social communication and social interaction. And as this overview here shows, I cannot point, right? TV screen, right? Yeah. As the figure shows, there is actually there is a high comorbidity with several other disorders, including intellectual disability and epilepsy, among many others like sleep deficits, anxiety, hyperactivity, attention deficit disorder, etc., etc. Autism spectrum disorders actually occurs in about 1% of children. The recurrence of autism in siblings, it goes up to 5 to 15% compared to the general population. So this increased risk of 5 to 15% compared to 1% really tells us that there is a very large genetic component running in these families. Let's say an aggregation of various studies performed to estimate this component, this genetic component, it was estimated around 80%. More recently, some numbers actually go a little bit down to 60, so between 16 and 80%, we believe that there is a genetic component in autism. Of course, then, if there is such a high irritability, right, to explain autism, can we actually find variants, genetic variants that contribute to this risk? And the answer is yes, and we can find them in different flavors. So we can find variants which we call rare because they are found in the population at the frequency of 1% or even less. Many of them are actually extremely rare, and they could even seen in one individual and almost not seen in more than 120,000 controls that we have now access to. And so these are the very rare risk variants. And of course you can say, well, if they are so rare, how many individuals can you see in the population that have at least that you can see the same variant at least twice? So that's actually very low probability, which means that to find, let's say, rare variants that would have 0.01% frequency, we would need to study 10,000 individuals to observe it at least twice or 3 times. Now, we do need this statistical power so that we can declare a variant as pathogenic, right? So there has been a really very large amount of work to be able to do that. Now, what we, in addition, find is that these rare variants can aggregate in pathways, and I will show you what type of pathways we see as affected in autism. So that is about rare variants. Now, we also would like to find common variants, meaning that variants that have a frequency of larger than 5 or 10% in the population, which means that we could, let's say, start to predict or go into personalized medicine to develop target drugs that would focus on this specific common variants, right? They would be common in at least 10% of the population. That would be the ideal situation, right? We would always go for common variants versus rare variants. Now, what we see is that in autism, they are very hard to find. As you see, I point there, the odds ratio is actually 1.2, which means that the effect of each variant is very, very little, right, alone. Which means, again, that we need to study many individuals, like 10,000, 20,000 individuals to be able to have the power to find these common risk variants. So, so we have tried to devise other ways on how to study and to tackle these common disorders that are very complex genetically. And so we decided to start to look at subsets that would have an increased homogeneity. Instead of just looking to all the autism with all these different comorbidities, we started to look, why not focus on a comorbidity that is seen, that is very prevalent in the population, like, for example, epilepsy. So in fact, we can see that epilepsy in autism can occur in up to 20 to 35% of autistic children, right? In other words, if you have 100% of autism, 50% of them, 60% of them can have intellectual disability, and about 35% of them can have seizures, right? So it's best to study not all autisms, but to focus on subsets that have an increased homogeneity, and therefore we would be able to have more power to identify those risk variants. And that's basically what we are doing. All right, so this is also to show you, actually to give a comparison of autism to other psychiatric disorders like schizophrenia on the side, bipolar, ADHD, even compared to celiac disease and anorexia. And basically what we are comparing here is the lifetime prevalence against the irritability. So you can see that autism is as irritable as schizophrenia, and even more than celiac disease or anorexia, right? So this is, or bipolar, or even type 2 diabetes. So this is telling us that if the genetic burden is high, we should be able to find these variants, right? Okay. So we have studied a very large number of families, and this is really the result of studying more than 2,000 what we call nuclear families, and these are composed of both parents, the affected child, and an unaffected sibling, and often even affected sibling. So, and by doing that, in this particular case, we find rare genetic alterations that can take different flavors, as I was telling you a little bit in a few minutes ago, which could be deletions, and deletions would be segments of the DNA that are lost. They could also be duplications, so segments of the DNA that are duplicated and inserted back in the DNA. And it means that if they insert in the wrong place, they can disrupt genes. If they delete important genes that have important functions in brain development, it means that we are going to have problems in neurodevelopment. And I'm talking of segments, really like large pieces. Now we can also have what we call single nucleotide mutations. So they only affect one nucleotide. So now, of course, we have these type of variants throughout the genome. Everyone has it. I also have it. Everyone in this room have it. Now, what happens is that in autistic children, it turns out that these variants are located, let's say, in the wrong place. Yeah, so they are placed in genes that have very important functions in neurodevelopment, And therefore, when these variants are there, they disrupt these functions. Sometimes they disrupt the function, sometimes they create a new function, what we call a gain of function. Either way, they are not the normal or the typical function of that particular gene, right? We don't want to have it more or less, we just want to have about the right. And sometimes these differences are actually very mild, but it turns out that some individuals have more than one. And so this burden is accumulated and creates a specific risk, risk profile for a particular person. Yeah, so each person has a different risk profile for, I'm talking about autism, but you can also talk about diabetes or any other disorder. Yeah, keep that in mind, that it's about the risk profile. Often it's not just about one mutation, it's about the background as well. Yeah, so you have the background and then you have one that has an increased risk, and it's the combination that really results in disease. Okay, in this particular case, as you can see, all these genes are affected in autism, and there is different classes of genes here. So some of them are copy number variants, so deletions or duplications. They can be loss of function, single nucleotide variants in these particular genes, or they can occur in what we call already known autism genes. And because the known autism genes, we know in which pathways they play a role, by analyzing them together, we are able to, by analyzing them together, we are able to infer what pathways are enriched for rare variants. So what pathways are enriched for genes that confer risk for autism, like in this particular case. So we see that there is a series of genes that are enriched for neurodevelopment and axon guidance, that you see here in yellow. So all these genes participate in those pathways. We also see that there is many genes that are enriched for functions like chromatin modification and transcription regulation. So this basically means that are genes that regulate other genes, right? So this is extremely important because there is a consequence. And then also others can have a more, let's say, specific effect, like they act in specific signaling cascades like the MAP kinase. As a whole, when we do, so this is when you incorporate deletions as well as single nucleotide mutations, yeah? So in other words, individuals with autism can have rare variants. These rare variants can be seen in only one or two individuals, but as a whole, so they target different genes, but it turns out that these genes, they participate in or they converge in similar pathways. So suddenly, we don't actually have to target each gene by itself, but what we are hoping is that instead of targeting each gene, developing a drug for the gene, and developing a drug for the pathway. See, so this is where we want to go when we deal with rare disorders, or disorders where the genetic architecture is made more of rare variants. Yeah, because they are all different rare individually for each gene. All right. So now I apologize. I was changing and these things moved, but so what we see, and that's also to reinforce why it makes sense to study and to use the same approach in epilepsy, is that the same genes, or often the same pathways that we see disrupted in autism, we also see them disrupted in epilepsy, especially what we call the pediatric epilepsies. So not the ones that start in adulthood, but the ones that can start in childhood, infantile period, childhood, adolescence. So these are the ones that have a higher genetic burden, and they turn out to have a lot of sharing or a lot of commonality with autism. So so we believe that this means that it's not so specific for the disease in itself, but it's just telling us that all those genes are very important during development. And then depending on the mutation and where is it located, you can have autism, or you can have epilepsy, or you can actually have both. And I can give you an example. SCN1A is actually a gene mutated in autism, mutated in epilepsy. Different mutations give autism. So loss of function mutations give autism, gain of function mutations give epilepsy. Same gene, but the mutations are just located in different places. So now, by knowing this, we learn more about the mechanisms, about the biological insights, about the role of these specific genes, and we are hoping that by doing that, we can develop better drugs with less side effects or more personalized. Okay, so we did the same approach in rat. Specifically, we focused on individuals that have rat-like features. And this is a syndrome that has actually a very high prevalence of autism as well as epilepsy. And there are classically 3 known genes mutated: MACP2, CDKL5, and FOXP1. So those are the ones highlighted in blue here: MACP2, CDKL5, and FOXP1. So that's what all we knew when we started this study. And then we collected individuals that had rat, but they were slightly different, meaning that when we screened for mutations, for these mutations in these 3 known genes, they had nothing. No mutations seen in the 3 known genes. So when we did whole exome sequencing, we find a variety of other genes. In fact, in 19 families that we studied, we found mutations in 12 different genes. So for us, it was hard to interpret these results until we asked the question, are these genes correlated or interacting with the known genes? And that's where this network comes along, right? These are, each node of the network is a gene, and the lines are the edges. So they signify basically interactions between these 2 genes. And we can see that all the other genes that we identified, they actually interact with the known genes. So this is telling us that they, even if it's different genes again, they converge in similar pathways, and therefore they explain why they have very similar phenotypes, very similar results in terms of phenotypic alterations. All right, so we are currently doing a very large study in collaboration with Patty and Steve Wolf here, which I believe they talked earlier. And so basically what we do, we collect blood, right? We collect blood for what we call the trio, so both parents and child, and also unaffected sibling that serves as control. And basically then we perform whole exome sequencing, and we look for mutations in these individuals. And then when we find the mutation that is where we have enough information to declare as pathogenic, these are delivered to the clinician. The clinician will discuss with the genetic counselor to better advise the individual that's going to receive their result. And then in addition to that, because several of these autistic children that have epilepsy, often they have what we call, they are drug resistant, which means that they are candidates for epilepsy surgery. That tissue that is removed during the surgery, which otherwise would be discarded, we can use it for research. And we use it to validate, then to look at in situ to see if the mutation is not only present in blood, but is also present in the brain, and if it's the same type of mutations. And then also how do they express, how do they distribute in different cell types. By doing that, then we can, let's say, accumulate more knowledge, again, to see how these pathways work in situ, in the cells themselves, right? This is not something that we cannot do with blood, because blood is not brain tissue, right? So this is really a huge benefit. And we can only do that in individuals that have this type of surgeries. Obviously, we're not going to do a resection in an individual that is not a candidate for surgery. I will skip this. I thought I had an hour. So this is actually how many families we already collected so far, and we are screening at the moment. This is about 367 families where the proband has autism and epilepsy. Yeah. In addition to the families that we are collecting, we also have access to very large data sets of autism alone and also epilepsy alone. So the idea is that we find mutations in autism comorbid with epilepsy, and then we can contrast with the mutations found in autism without epilepsy and epilepsy without autism. And we can look into the mechanisms. All right, so this is just to give an overview of some of the findings. We are finding mutations in GABA2, which is a GABA B receptor subunit. So basically produces a neurotransmitter, and there is also some studies in mice that back up the same type of phenotype that we see in humans. And we also see mutations in a galactose transporter, the SCL35A2, and this is kind of interesting because it means that metabolically, so when you do a metabolic test, you could actually find an alteration in the levels of this galactose transporter. So this opens new avenues to start to screen for alterations in serum, in plasma, so basically to go into the metabolic alterations that otherwise we wouldn't. Okay, so now I would like to pinpoint on the Landau-Kleffner syndrome. And this has been interesting because we find that some of these individuals with LKS do have autistic features, not all of them. But we place LKS in really what we call a lateral clinical spectrum. And really here you can appreciate that there is a severity gradient where you can start from benign pediatric focal epilepsies, and here is benign because the course of the disease is considered benign, and they can get free of seizures very early age, about 2 to 3 years of age. And then Landau-Kleffner syndrome is much more severe. Typically has language regression, which can occur around 5 years of age. And 20 to 30% of individuals do not have seizures, in fact, so they only have an EEG abnormality, so an abnormal electroencephalographic profile. And then we, in this continuum, there is the other one, the syndrome that we classify as continuous spikes and waves during sleep. And there, in addition to language regression, we can actually observe global regression, right? So as you can see, so you start from the benign, where there is almost no regression. You can have LKS, where there is regression of language, mostly. And then CSWS, where you have a global regression. So at least 2 domains will be regressed. So typically with neurocognitive decline. Now, this was important for me to describe this because we have been studying a specific family, and it was important for us to classify and to look at the progression of these different features, so language versus other domains, to compare to genes that have been already identified as mutated in somewhat similar phenotypes. So we started, and this was really, actually typically you start by looking at the literature and coming up with a list of candidates and then screen for these candidates. Here, we turn it around, so we actually do what we call a genotype-phenotype approach, where we just started with the whole exome sequencing. We had no idea which gene could be involved. And with whole exome sequencing followed by Sanger sequencing for confirmation in the whole family, we verified that UBTF, which is a gene that codes for a regulatory of a very important enzyme that regulates transcription of other genes. And it turns out that it is mutated in an LKS individual. Over here. So typically you would expect a C, and what you obtain is actually T. Yeah, so this is what we call an heterozygous, so we have both C and T on the particular exon 7 of this gene. And this is another version of the same. Now, what became very important is that this mutation is seen in the boy, in the patient, but is not observed in either of the parents, neither the sibling. Right, so this is what is very rare also. We do not see it in more than 120,000 individuals of, let's say, general population. And it changes glutamic to a lysine. This is what we call missense mutation, and is basically very conserved and disrupts this domain that basically it's very important for transcription regulation of other genes. Now we are trying to find out more about the function of this gene and why this mutation is so important. But as we find this mutation, then we went through the literature and to check whether it has been published. At the time, not, but a few months after, 2 other papers came along, which are these 2. And now the interesting thing was that while the individual with LKS only has regression at a language level, so there is no neuro regression specifically per se, so there is no neurodegeneration, there is no cortical atrophy in the individual. We do see the same mutation as in 11 other patients that do show neurodegeneration. So this brought us to study this a little bit more in detail and try to compare between these 2 studies. I think this is my last, yes, last slide. And I thought that this was important to notice because between these 2 studies, you see, so there is the Toro et al. They found 4 individuals, 2 males, 2 females with that mutation. The other one in 2017 finds 6 females and 1 male. I can tell you that to find such rare variants in 11 patients, that's quite a finding, really, because we typically find mutations in 2 to 3 individuals, maybe at the maximum 5. Here we find about 11. In this case, now 12 with the individual LKS. So my view and my take on this is that this mutation, first thing, it's very important to screen this mutation for any individual that would be a candidate for neuroregression. That's to start. But second, I think that we might start to think that it might be also associated to more mild phenotypes, right? Not only individuals that have neuroregression with neurodegeneration or loss of cortical tissue, but also in individuals that only have regression of one domain, right? So this really opens the door to start to study this gene and other genes that have similar functions in LKS. So at the moment we are collecting larger sample sizes. We actually screen already in 8 other individuals with LKS, and we did not find it. So for now, this is the only individual with LKS that has this mutation. So this is a call for everyone. So if you have LKS patients and you want to participate in the study, please get in touch with us. We would love to get you on board. All right, so. It's just to thank everyone in my lab and also the consortiums that we work with. Thank you.
Thank you very much, Dr. Pinto. It's been very interesting.